I'm Kathy Turner. I am sort of the technical person for these webinars. And with me is Robin Bowen, who's one of the members of the Breeder Education Committee, and Ann Hubbs, who is the chair of the Health and Genetics Committee. So I'm going to be like a little square up in the corner and learning like the rest of you. So with that, Robin, I'm going to ask you to take it away.
Right out of the gate. It's something that I've become aware of lately, that a lot of people believe if they get a DNA panel done, they have full clearances. How does that fit with the code of ethics?
Oh, I think that's kind of what we're going to be talking about today, isn't it? Yeah.
Yeah.
Yep.
Yeah.
That's not a clearance. And in fact, it's more of a test. And there are ways that dogs can be a carrier, and it's fine to breed them. Very different than what we think about in terms of our clearances. So maybe we should talk about all of that. Do you want me to start the talk?
Yeah.
Let's do that. So can you see it now?
Yes.
Okay. So we're going to talk about DNA health testing in golden retrievers, and we're going to talk about what's clearance and what's DNA health testing. And I wanted to start with a couple of disclaimers that the webinars that we give are addressing current canine diseases and are intended to provide general information from experts on that topic. The opinions expressed are those of the speakers and are not necessarily endorsed by GRCA or any other organization. Importantly, your Golden's veterinarian provides your Golden's veterinary medical care. We do not. So please check with your Golden's veterinarian before giving any medication or treatment. A veterinary client-patient relationship and access to medical records are essential components for veterinary medical care. So thank you for joining us today. This talk is largely because we were getting some frequent questions, and the Breeder Education Committee was kind enough to ask me if the Health and Genetics Committee might have some answers to these questions. And we'll start with the answers and then an explanation of why. And the first question is, who makes the decisions about the GRCA Code of Ethics? And the answer to that is it's the GRCA Board of Directors. But there's always input from the membership and from relevant committees. So what is the role of the Health and Genetics Committee in terms of DNA testing? And the Health and Genetics Committee informs the board and releases advisory statements to the membership as new DNA tests become available. You can see some of those statements on the Golden Retriever Club of America Facebook page, or you can go to the health section of the GRCA webpage, or you can read Golden Retriever News, our official newsletter. So what is the composition of the Health and Genetics Committee? And the answer is currently there are 8 veterinarian members, and there's 1 research facilitator, a non-veterinarian breeder, who is very important because some of you have occasionally seen me start talking and all of a sudden I'm talking in a language no breeder would ever understand, and I've messed up, and we always have to have a layperson bring me back to reality when I do that. So please do if I do that in this talk. So the real question that we started with today was, why does the Code of Ethics, our COE, specify that clearances for hips, elbows, eyes, and hearts of breeding dogs need to be online in that database that is verified by OFA? But it doesn't say anything about DNA tests. And the short answer to that is that information on hips, elbows, eye, and heart status of siblings and ancestors really help us as breeders, and I'm a breeder too, to make decisions in terms of breedings, because there are multiple genes that affect those, and there is no validated DNA test. And so we want to know what the hip clearance is on that dog, and breeders want to be able to look at the pedigree and see how strong that clearance is. It really helps. And if we did not have those reports online, they would be very difficult to find, particularly if the breeder is no longer with us. For most of the DNA tests available today, recommendations from leading geneticists are simply to avoid breeding two carriers to each other. And there really is no need to know the status of the siblings and the ancestors. You could have an affected sibling and you could own the clear sibling to that affected dog. And it's the status of the dog you're breeding, not their siblings. If it's a autosomal recessive disease, which most of our tests are testing for. So here's some information that breeders may find helpful. And the first one is what the GRCA Code of Ethics actually says. And that is that the ideal use of DNA tests is to prevent producing affected puppies, while at the same time maintaining genetic diversity and gradually decreasing the prevalence of the disease genes in the breed. And tests are available for some autosomal recessive diseases today, and we're going to talk about those. And autosomal recessive diseases are those diseases that can occur when a puppy gets a gene for that disease from both parents. If either parent is normal clear, does not carry the abnormal gene, that means the puppies would not be expected to get the disease. They might be a carrier, but they're not going to get the disease. They are healthy puppies. And importantly, automatic exclusion of carriers of recessive diseases from the breeding population is not recommended by leading geneticists. And the reason for that is because genetic diversity is critical to breed health. And in fact, it is genetic diversity that for a breed as a whole tends to keep the breeding population healthy and prevent us from identifying diseases we never knew we had in our gene pool. But for the individual, it's important to test for some critical diseases, particularly those that are lethal and could kill the puppies. There is also a DNA test for one mitochondrial disease that can be lethal. It's sensory axonal neuropathy. That disease can be passed from a specific female line to some puppies, but it's rare in North American lines, and I will briefly tell you why. So some basics. Animals are made up of cells. Cell under the microscope, it looks like this, if you can see my pointer. So that's the cytoplasm. It's usually on the outside, and it contains the metabolic activities and critical organelles that make the cell function. You don't want it dying, you want the cell to live. And the critical organelles include mitochondria that provide chemical energy for the cell. And we talked about SAN, okay, sensory axonal neuropathy. Mitochondria come from the dam. They're not from the sire, not at all. And the reason for that is because SAN is in the mitochondria out here, like this. And that would be in the egg cell if it's from the mom. And when the egg is fertilized, the sperm has a tail. That gives it its energy, and that's how it moves. And it has all the mitochondria, and it falls off at fertilization. So it doesn't get in there. So all your cytoplasm, well, most of it, comes from mom, and all the mitochondria come from mom. So the only way you're going to get SAN is if you have a puppy whose mother's mother, and so on, goes back to a golden retriever born in Europe in 1971. If your bloodline has been in North America since the 1960s, and you can trace that female line, the bottom part of that pedigree on down, and you get to 1969 and you're still in North America, you are home free. So you don't need to.
But what are the symptoms of SAN, Anne?
SAN is a disease where the puppies, depending on how the number of abnormal mitochondria they got from their mother, because they're not all the same, they will begin to become ataxic, so they have trouble walking and they become weak. And that's what makes it so difficult, is some of them have enough normal mitochondria that they don't have any signs of the disease, so they appear perfectly healthy. But if they have, say, 25% of their mitochondria abnormal, and in forming one of the eggs, a lot of those mitochondria go into that egg, that the individual that results when that egg is fertilized may have this progressive neuropathy. So that's a problem. And it can be fatal. It can sometimes get to the point that the dogs can't stand up, and we have to put them down. Now, the nucleus is what we're going to talk about for the rest of this talk. And it contains DNA inherited from the sire and the dam. And it is, in fact, in the nucleus where we have all those nucleic acids that we talk about normal inheritance. And it's right there. Now, what we really care about in the nucleus is the DNA. And the DNA is kind of a giant zipper that is wrapped around little pieces of protein. So it's this giant zipper, and in fact, it can come apart in many, many different places. So it's a string. These aren't jelly beans. When we're talking about genes, they're organized on a string, and there are other genes that are, of course, they're neighbors, and they often go together. And that's important. We'll talk about that a little more in a minute. Now, dogs have 78 of these zippers, these strings. We call them chromosomes, and they're arranged in 36 pairs. The reason they're pairs is you got half your chromosomes from mom, half of them from dad. So your dogs, they've got 36 from mom, 36 from dad. And that's how you end up with 78 chromosomes. Now, one of each pair is from the sire and dam, but one pair of those chromosomes determines the sex of the puppies. And we call those the sex chromosomes. Now, females only have one type of sex chromosome. It's the X chromosome. They've got two of them. Since both of their sex chromosomes are X's, and they're going to pass one to each puppy, one of their two X's, oh, all their puppies are going to get that X, right? Because they only can pass down an X. They don't have a Y chromosome to pass down. So they only pass down that X chromosome. So all golden retriever eggs come from mom, and they contain an X chromosome. Males have an X and a Y chromosome. So they could pass down the X chromosome, or they could pass down the Y chromosome. And about 50% of the time they do an X, and about 50% of the time they do a Y. And so as a result of that, if the sperm containing the Y chromosome fertilizes the egg, then it becomes an XY, and it's going to be male. And if the sperm containing the X chromosome fertilizes the egg, the puppy will become a female. And that's the end of today's talk about the sex chromosomes. What we're going to talk about for the remainder of the talk is the 35 pairs that are known as autosomes, all the rest. And genes are on those chromosomes, those giant zippers wrapped around proteins. And the genetic code of each gene is made up of a pair of nucleotides in a specific order. That order and that code is transcribed into something called RNA. That's important because RNA is translated into protein. The proteins are actually doing the functions in the cell. When we're talking about genetic abnormalities, the concern is if the gene is coding for something that is translated into a protein that can't get the job done. Proteins determine how the cell works, so that's a problem. The DNA code is actually the language of health and disease, and this is what it looks like. It's a sugar phosphate backbone, goes around like this. And on that backbone are base pairs, adenine mixed with thymine, guanine with cytosine. Sometimes it would be adenine with thymine. And the zipper unzips, and the code is read by RNA, and it gets translated into protein. And that's the difference between health and disease in many of these conditions we will discuss. Now, it's important to realize that codes may vary without causing disease. In fact, that's very common. I don't look like you, and you don't look like me, and different dogs look different. And that's because there are genetic variants. They cause differences in appearance and sometimes differences in health in dogs and in people. In people, we know that each person has millions of variants. We don't worry about variants unless they cause disease. It's what makes the world wonderful. We're all unique. So in this case, in this example, which comes from a human genetic disease website, which has great illustrations and is in the public domain, you can see that cytosine in one person may be thymine in another. It doesn't matter as long as it just makes them different. And here's the website. But some genetic variants can cause disease. And single gene disorders are caused by variants at a single site in the DNA. And for recessive single gene disorders, which are those disorders we test for most of the time, an affected individual receives the variant from each parent. And the products of the gene are different, but the cell just needs one normal copy for normal function. So it doesn't matter with a true recessive if they've got a variant gene, as long as they're just a carrier. They're perfectly healthy. But if they don't have at least one normal copy, disease can happen. Again, this is from a human site. But if you know someone with cystic fibrosis, what happened is they got a critical variant from both mom and dad, and it's on human chromosome 7. And because of that, they can't produce a critical protein in their lung, and they therefore develop the disease cystic fibrosis.
So the normal gene is the one that continues to do the proper work of that gene. And
In someone who's a carrier, if it's from both mom and dad.
And so without the normal gene to do that work, that's when we have disease.
Yes.
Okay.
Yeah. With these recessives.
Yeah.
So to make it kind of simple, we have something that's called the Punnett square, I think is what they call it. But what we do is we look at these genes as being normal, capital N, or not normal, we'll make it a little n. And we can figure out what mom and dad would do, kind of like the X and the Y. So if you have a carrier bred to a carrier, so they have a normal gene and a not normal gene, and you're breeding to the same thing, another carrier over here, they have a normal gene and a not normal gene, then we can break these two possibilities, 50% chance of each. So they could send down a normal gene or the not normal gene. And the other one, they're a carrier too. They could send the normal or the not normal. We made the not normal red here, so we all pay attention to it. And there's the problem down there. If both parents are carriers, you got the two not normal genes together and you'd have an affected puppy. So in this situation, this is what we want to avoid, is if we have two carriers for a significant autosomal recessive disease, each pup has a theoretical chance at conception of being 25% normal clear, 50% chance of being a carrier, and 25% chance of being that affected puppy. And that's what answers that original question, why do we not have to put all these DNA test results out there on the OFA database? You can if you want, but you can have a normal clear puppy over here and its sibling, maybe that one that's affected. We care because we're worried about that affected pup, but we don't have to know that that was the uncle. But it's a little different when we get into some of the diseases where we actually do phenotypic clearances. So let's do the same thing and let's see what happens if we take a carrier. Maybe they're the best dog you ever bred, but they're a carrier that has one gene for something that's not normal, and you're breeding to normal clear. Are you going to have any sick puppies? For heaven's sakes, no. You just got this great best dog you ever bred, and half of the offspring are clear and half of them are just like the best dog you ever bred, and they're all healthy puppies. It's wonderful. It's good. You might want to tell people that they've got a carrier, or they may have a carrier. It's important that we are transparent. Puppy's going to be healthy, but if you breed it, you don't want to breed it to another carrier. No pup is affected. So what are some of these DNA tests that we have in golden retrievers? And could we just go and grab any DNA panel and they say they tested for 270 genes and your dog is entirely normal, they must be the best golden retriever ever born or bred? Well, it doesn't quite work that way. There are a few that are really important. The big one, neuronal ceroid lipofuscinosis, golden retriever form. It is common in some lines. It's not common in some other lines, but it's a progressive neurologic disorder. Puppies seem normal at birth. They are normal when they go home with their owners. And if you send a puppy home that has this disease, you are sending a puppy buyer home with a puppy that is doomed to die as a young adult from a slowly progressive disease. Do not do this. Please don't do this. I will personally feel terrible if you do, and you probably will feel worse than I do. So please don't do that. Test for this one. Golden Retriever Congenital Myosthenic Syndrome, Golden Retriever CMS. You won't see this on any of the large panels. It's a progressive muscular disease. We believe it is rare. It was described last year for the first time in 4 Golden Retriever puppies from one breeder in North America, and we don't know how common it is. It is lethal. It will kill your puppies. It is not on any of the panels. There is a test. The only place I know that's currently offering it is University of California at Davis. We do not know how common it is. They are testing more and more Golden Retrievers, and we will find out how common it is. But it may be very uncommon in the Golden Retriever gene pool as a whole. But if you're line breeding something different than everybody else is, and many of us do that, it might be in your line. So you don't know.
So that test is not on most of the commonly used companies' big panels, because UC Davis hasn't published it yet. Is that correct?
No, that UC Davis published it.
Oh, they did. Well,
Yeah,
then,
you can find the reference to the publication on the health website. Yeah.
okay, so
They published it. And in fact, I wrote to a couple of the big companies and said, are you going to be testing for this? Because the code, the sequence is published.
right.
And they replied to my email.
Interesting.
I got no reply. I get no response.
Interesting.
I was like, hey, this is
The
illegal.
work is done. Somebody
The work
found the location.
Here's the publication.
Yeah.
Oh, whatever. Golden Retriever Ichthyosis 2. Now this one's a little bit different. That's the one where the mutation is not yet published, but we know it's been identified. They're working on the publication. This one, the only test I'm aware of for this particular disease is offered by the University of Pennsylvania. They have not published what the mutation is, and we have informed the membership that we have several breeders who have used the test, and it has helped them to eliminate the disease simply by avoiding breeding carriers to carriers. So here it's kind of word of mouth, but we've also talked to the university, and it's a leading academic research institute. They are offering it, and they have told us that the disease can become unmanageable, and some of the dogs have had to be euthanized. And we like to be transparent too. So there is an article, you can also find it on the health website of the Golden Retriever Club of America website. And just go over to the health section and you'll find a section on Ichthyosis 2 and the test. You can do the test if you want to see if your dogs may be carriers.
Let me interrupt. One of the people watching says that CMS is on the Embark panel.
Oh, is it? Okay.
And somebody
But
else.
is it Golden Retriever CMS?
I don't know.
Because they had a CMS on there and it wasn't Golden Retriever CMS.
Oh, okay.
But they may have added Golden Retriever CMS.
And then the Ichthyosis 2 is PenGen.
PenGen, yes.
Yeah, just ask for a repeat there.
Yeah, and they may have added, the last time I checked.
I'm looking it up while we talk.
And the last time I checked, it wasn't the Golden Retriever CMS, but they could, it's published, and these big companies do web searches and they can pick it up, and they may even look at the Golden Retriever Club of America website. I don't know. It could be. Yeah. PRCD, PRA is one of the first gene tests ever developed, and it causes an adult onset progressive loss of vision leading to blindness. Depending on the individual and the breed, it can be fairly late onset, which is good, but can be prevented by avoiding breeding two carriers together. Golden Retriever PRA1, it's also typically an adult onset progressive loss of vision leading to blindness, as is GRPRA2. These are fairly widely available, I think, at this point. Golden Retriever Ichthyosis is one that's a little bit different. It is very common. In some countries, less than a third of the Golden Retrievers are normal, and the majority of the Golden Retrievers are affected. And the normal appearance of this is puppies having dandruff, but adults being clinically normal. However, they can develop significant skin disease. And we know the gene is responsible for the keratin layer of the skin. So in a retriever breed, we'd like to see this gene frequency reduced. But certainly, we do not want people to try to discriminate against the carriers of this disease, because that would be a huge problem for us as a breed, because it is that common of a disease. But it's a useful test. So if you're trying to decide between two equal potential mates and one of them doesn't carry the gene and you're breeding a carrier, you are less likely to have significant skin disease in the puppies. And certainly if you're going to be having them swimming around in murky water, you might want to worry about it.
Anne, what is the likelihood of being PRA affected?
For a puppy that is from carrier to carrier, it's 100%.
Oh, yeah.
It varies with.
No, I mean, what are the numbers looking like? Do you know?
It's the affected. Optogen used to be the only one who ran this test, and they've been bought by the Wisdom panel. And it used to be Optogen would send me reports of the prevalence. Now, the big companies won't do that unless you buy membership, and then they get to keep your DNA. And so, yeah, that's a different topic entirely. I don't want to get off on that. But the last report I got from Optogen before they were acquired, we had seen one actual case of PRA in the previous 3 or 4 years. Now, they had a lot of positive carriers. But people knew about the test, and that's why you don't see us saying everyone should test their dog for PRCD, PRA, but we say it's a disease out there. If you don't know whether it's in your line or not, by all means test. But if you've had the line and you've tested their ancestors and they were negative, you may not want to run the test. I would run one of the big panels. They all have these. And then add in the other test for serious diseases. But one of the issues with the big panels, and we're going to talk about that in a minute, is they test for a lot of diseases that aren't in Golden Retrievers that can really confuse people. Like, are they offering a CMS test for a different breed?
It appears they are offering the Labrador Retriever CMS in my quick study here.
Yes,
but
yes. And there
not Golden Retriever.
Yes, there is a Labrador Retriever CMS test, and it will not pick up Golden Retriever CMS because it's a different mutation. And that's one of the problems with the big panels, is someone can easily make a mistake. There is another one shows up on a lot of panels. It is a golden retriever dystrophic epidermolysis bullosa. It's been described in one line of European golden retrievers. And if you want to test for it, it's mild epidermolysis bullosa, but hey, you're probably going to have a dog that's clear. It's fine to test for it, but it's not like the importance, of testing for neuronal ceroid lipofuscinosis, where we know NCL is common in some bloodlines. And that and the golden retriever CMS, you're going to be sending home a healthy puppy with someone who's going to watch it die because you didn't do a test. That's bad. So we keep talking about this genetic diversity thing and why does it matter and why does it reduce the risk of recessive conditions? And here's why. Okay, so if you have a gene pool, it's over here. Each color ball represents a dog that is a carrier of a specific disease. When it's bred to a dog that is a carrier of the same genetic disease. And in this representation, each of these little balls is a dog with a genetic disease. And if these two come together, they're going to get pink disease. And over here, that's orange and red. Let's see. Well, we're not going to get very many diseases over here in this diverse gene pool. This may be the original population of golden retrievers, but then some popular sires come along, or there's some other constriction, like that original golden retriever gene pool got imported to North America. Now, once these get imported to North America, unless you bring these back, doesn't matter how many golden retrievers come out from the other side of the bottleneck. You can't get back the purple genes you lost back here. So this is the problem with bottlenecks. And this is how genetic diversity protects against the disease you can't test for today. And I guarantee you, there are far more diseases we don't know about that can affect our dogs than the ones we do know about. And if we add in another big constriction, a bottleneck, let's say, let's get rid of all the dogs with Ichthyosis 1. Let's just breed the dogs that are normal clear. Well, then you can actually use a genetic test to be a bottleneck. We don't have to do that. We can prevent any puppies from being affected. Other things can cause the second bottleneck. So you start out here with very low risk. You come to a bottleneck, you get higher risk. Other bottlenecks, you got to have a coat 10 miles long in order to win in the breed ring, whatever it is that causes a bottleneck, you got big problems. And the reason for that is now it's very easy for two dogs with the same genetic disease to be selected as breeding partners. In reality, it's probably a little bit worse because every dog probably carries multiple recessives. So if I'd been able to draw multicolored circles, some of these dogs would have been carrying yellow and red and tan. So it really matters. We want to avoid that bottleneck. There's another situation too when we start really heavily discriminating against carriers. Doesn't mean you have to breed your carriers, but don't just say, let's get rid of all the carriers. That would hurt our breed. That makes it worse. But the other thing to remember is many genes are on each chromosome. It's not jelly beans. I made them balls, but they are not jelly beans. These chromosomes are in a line. The genes are lined up on those chromosomes. And that goes back to what the Code of Ethics is saying. The ideal use of DNA tests is to prevent producing affected puppies, while at the same time maintaining genetic diversity and gradually decreasing the prevalence of diseased genes in the breed. Genes located close to each other are said to be linked. Dogs have about 20,000 genes organized onto their 39 chromosomes. No.
And so when things are linked, they tend to be inherited together.
Absolutely, absolutely. So when the egg and the sperm meet, the matched chromosome pairs, one from the sire and one from the dam, they actually exchange some genes. They've got all those genes, they're lined up on those 2 chromosomes, and they start throwing strings of genes across as groups, as clusters. And the genes that are very close together tend to stay together because they're linked genes. Multiple genes can be linked to the gene identified in the DNA test. So
What does that mean?
what that means is they are going to stay together so that when you say test for, we'll use Ichthyosis 1, the gene that's next to it on the chromosome has a very low chance of not being associated with it. So it will tend to stay right there, because when there is this exchange of genes, it's like trying to mix up a bunch of teenage kids in a group. Some of them like each other and they stay together. It's the same thing with the genes. And the geneticists actually have mathematical formulas. And at one point I was trying to figure out what the chance of 2 genes that were the neighboring genes on a chromosome, how many generations as a rule would it take before it was probable that one of those, that those would be separated? How many breedings would have to be done before, as you get further away, it's more likely they will go to the other chromosome? And the geneticist did the math for me, and he said, well, you'd have to do an average of 14,000 breedings before you would likely have those 2 genes show up in one individual in separate individuals. So it's not that they can't be separated. There is this exchange of genes, but they go in clusters. They grow in groups like those teenage kids. So one of the things we have to understand is some genes close to the disease genes are likely to be important. So what if a rare variant next to one of them could prevent hemangiosarcoma? That could happen. So if you decided we want to get rid of all the carriers, and we know that some of the cancer genes are going to be described probably in the next 5 years. We know there's some on chromosome 5. We know in the flat-coated retriever, there's some on chromosome 19. And we start fiddling around with getting rid of our carriers and the adjacent genes, we might be getting rid of that 10% of the population that didn't have a cancer susceptibility gene. We don't know how.
Because they travel together.
They travel together.
So you get rid of this, you get rid of these things you don't know what they are good for.
We have sequenced the canine genome, but we don't know what all those genes are. Yes. And let's not get rid of genes right now, because we know very few of the genes that are going to be important. And the conclusion is rapid elimination of carriers from the gene pool can reduce diversity of closely linked genes, even if their siblings are used in a breeding program. Because those ones that we're excluding because they're the carriers, if we were to do that, and we don't recommend that, if we were to absolutely exclude the carriers themselves, we're also going to exclude many of their closely linked genes, even if we use the siblings. Now, over many generations, it's more likely that the exchange of DNA at some point is going to be very close to the variant. But you want to do it over generations, not immediately. But you have to do it responsibly, especially if you're talking about a lethal disease. I mean, if you're talking a lethal disease and you don't tell a puppy buyer that their puppy could be a carrier from it, shame on you. That's terrible. It's just not right. We all know that's not right.
Well, there's a whole group who believe all NCL carriers should never be bred.
And I have tremendous respect from some of the people that believe that. However, the geneticists have repeatedly told us, just be responsible. make sure that the carriers are not bred to other carriers. And there is more responsibility in producing those litters. But from a veterinary standpoint, is the risk of that, and most of the people I know who are dealing with serious genetic diseases in any dog breed, they're going to place those puppies with personal friends.
Mhm.
And the risk in that scenario where you're not just screening a normal puppy buyer, you're talking people you've known for years. They're next to family. The risk from there is far less than the risk that the dog is going to fall into bad hands and be abused. An owner dies, it ends up, they forgot to have a will. I checked a draft will for myself at one point, and the lawyer had drafted it with everything going to my son's special needs. Every possession would go to my son's special need trust. And I said, what would happen to the dogs? And he said they'd be auctioned off for the special needs trust. And I was like, change this will. But you can imagine, I read the fine print.
Yeah.
There are people that don't. Their dog could be auctioned off. Yeah. If you don't read that will, it can happen. And the important thing is we do not know the function of many genes in dogs. So while I absolutely totally respect people who would never keep an NCL carrier, if someone can do it responsibly, there may be genes that are really important that are next to that. I don't know. Yeah, we just don't know. And I wish we did. But the critical thing is to prevent bottlenecks using DNA testing wisely. And by wisely, the important thing is to grasp that we can only test for a small fraction of the genes important for golden retriever health. And we've got to use DNA tests if we are going to use them wisely. We need to prevent puppies from getting autosomal recessive diseases we know about today. Removing carriers from the gene pool can create bottlenecks that reduce genetic diversity. That doesn't mean we don't eventually want to get to getting rid of the carriers, but do it slowly. Check the phenotypes of those dogs, keep them around, be responsible. Do not allow two carriers for recessive breed diseases to be bred to each other.
And let me just be the person without, the knowledge. You said check the phenotypes. You do not mean
I'm sorry.
DNA.
No, phenotype is you're looking. One of the things that was really hard for me as a veterinary student, I'd sit there in class and I would just try to get my head around it. But one of the things that was really hard for me to grasp is that if we want to make a lasting difference in the breed, we have to breed a dog that is very successful at something.
Mm-hmm.
And if we as responsible breeders can't create a dog that is successful at something, that doesn't mean we haven't done something good. We've produced some wonderful puppies, but we won't help the gene pool.
Right.
So the phenotype is we're competing with these dogs and we're showing that they're very good. Now, that doesn't mean we don't have to take a dog that is just a normal golden retriever and healthy and sometimes breed it. We do. And we can breed those dogs. But when we want to make some of these dogs that have lasting impacts, if we as responsible breeders don't do so, then the breed as a whole will suffer because the irresponsible breeders will.
Mm-hmm.
So we have to do that's the reason for what the Code of Ethics says, which is the ideal use of the DNA test is to prevent producing affected puppies.
And on that, Anne, can you expand on whether people should be testing for CMS, golden retriever CMS and NCL, and should those be listed on OFA? If not, what should be listed on OFA within the DNA package?
We have not stated what should and should not be done for DNA testing in the Health and Genetics Committee, because as soon as we would say you should test for NCL and put it in the code of ethics, then CMS would appear and it would go on and on. So we haven't specifically said that, but we have said that all golden retrievers should be tested, that the Health and Genetics Committee believes all golden retrievers should be tested so that they cannot produce any NCL affected puppies. And if a breeder is comfortable that, we're not going to get angry at a breeder who has a dog that's the product of 2 NCL normal clear dogs and they don't test it. And, but because they've made sure that they're not going to produce any affected dogs. And the important thing is you can't get NCL unless both parents are carriers. Right. So if someone's breeding to a dog that's had an NCL test, they don't have to test.
So the people who own carriers for CMS and NCL, yes, they should list that on OFA, or would that be a more responsible thing to do? Or
I don't think they have to advertise it. I don't think even when we talk about online databases for hips and elbows and eyes and hearts, we only say that the normal results are put there. It's not a calling out of someone that produced the carrier, because we do see people that, you know, when NCL came out, there were people that were claiming it was my fault and were sending me hate messages and texts at 3 a.m. in the morning. Yeah, I knew about it when Ron Rubrick told me about it, and I'm very grateful to him. But they don't have to say their dog is an NCL carrier, because there's some people out there that you know, they may not follow the same rules I do, but we put things on the OFA database to say the dog is clear. So with hips and elbows, you can seek the healthy dogs. We don't ask that. I mean, I put my affecteds on OFA. I think it's great to put affecteds in the open database. It's transparency. But if people don't want to do that, I'm good with it. But they need to do what they need to do to make sure they don't produce any affected puppies. So if they don't want to test for NCL, but they're breeding to those that are NCL normal clear, it's okay. I think with NCL, I think it's a brave and good person that puts the carriers out there so those that have relatives will test. Mm-hmm. And I think we need to change from, look, they produced something bad, to, wow, that was really courageous.
Brave. Yeah.
Courageous, good. I'm so glad they said that, because I actually have a dog that's descended from the litter brother or whatever. And that's a good thing. But that's why when it comes to the code of ethics, we're not going to say, you know, test all dogs for NCL, because it's a what if. Are they breeding in them to a dog that's clear? And, you know, for some reason, they're having to send their kid to college and they can't afford it. Yeah, it happens. But there's another thing too, and that's laboratory quality control really matters. Do not low bid your DNA test, because DNA tests are only as good as the technician who ran the test. If they switched your sample, or if you collected the sample from the wrong dog, or if you mixed them up and you used the swab on 2 separate dogs and contaminated the DNA, there's all kinds of problems. So it's important to remember that DNA tests are highly accurate if they're performed correctly. Errors can happen if quality control standards are not met, and they are not cheap. And information on some of the laboratories and some of their credentials you can find online at this address. It's DogWellNet, and they actually have things like, are they part of international organizations? And that will help you with your selection. The other thing to remember in terms of quality control, and this is a really hard one to get one's head around, is the risk of an error in at least one test increases with the number of tests run simultaneously, particularly if the mutant gene is very rare in the population. And we know that from the human over-the-counter testing, where 40% of the false positives on the DNA panels, 40% of the positives have been reported to be false positives in the DNA direct-to-consumer tests. And you sit there and you stare at that number. So if you do a panel and your dog has a positive in a rare gene, if it were like the human genes, then it would be how likely it was a false positive? Well, I don't think we're as bad as the human direct-to-consumer tests. The reason for that is if you have a test that's 99% accurate and you do 1,000 of those tests, what are the odds that one of those tests will be wrong? And it's quite high. If the gene is rarely positive in the population, say the rate is 1 in 2,000, then the odds, if it shows up positive, are significant that it's actually an error. So just because you had 127 tests done doesn't make it better. It's not necessarily better because of that math, and also because if the lab is doing 127 tests, how carefully are they doing them all? And so you were asking about NCL. If I was breeding a normal clear to an NCL carrier, I'd probably have 2 labs run that test.
Yeah, and we have a question on that actually.
Okay.
What is a bitch owner to do if the stud
Yes.
dog owner is not transparent about NCL carrier status? Just move on to another stud dog if there is not a place to report the DNA testing?
You can go to another stud dog, or if you absolutely decided you want to breed to that stud dog and you've got this non-carrier female, I would test her twice.
Yeah. So when these companies that do the the 500 things,
Yeah.
genetic testing that are for dachshunds and whatever's, not even Goldens, the reason that they offer that whole thing is because of the nature of how they run that panel. They can't just pull out, or it's more expensive to just pull out the golden retriever things. It's cheaper to run the entire 187 or whatever conditions.
I kind of asked that question to myself, so I ran one of them on one of my dogs. What's this like? It's actually really cool. They tell you all these stories about your dog. I think it's marketing.
Yeah.
My dog was tested for 127 canine diseases, and my dog had none of them. Woohoo!
Yeah. Well,
I have the best dog in the world, right?
A lot of breeders who don't really conform to the code of ethics or fall into that trap, and they advertise litters, based on that.
and that's there's the panels have their place. I took the panel I did on my dog, and the parents had been tested for the common diseases by other methods, and sure enough, the panel said he didn't have those. But I will also do CMS and test him for that as well, and Ichthyosis too. And the important thing is to double check if you're using a carrier of one of the fatal diseases. The odds are 99% that there's not going to be a problem, but I don't want a 1% chance I'm going to produce something like that. So how do the DNA tests vary from hip, elbow, heart, and eye exams? And the DNA tests for autosomal recessive disease identify a gene that can cause the disease when it's received from both parents. Hip, elbow, heart, and eye examinations involve veterinary specialist exams for the presence or absence of the disease at the time of the exam. Those are believed to involve unknown or multiple genes, so they're polygenetic, and they sometimes have environmental interactions. For example, recent data on golden retriever pigmentary uveitis suggest it may be polygenetic. The online databases are approved databases that are registries under management of veterinary professional associations, registries maintained by nonprofit organizations with veterinary staff or advisory boards, and university registries under veterinary advisement. In the United States, that's the Orthopedic Foundation for Animals. It's the only one that meets those criteria at this time. And the importance of them is that provides documentation of normal examinations and the age of the examinations. And let's face it, it's the age of fraud. And that's one of the reasons for having that. And it helps breeders to identify pedigrees that may reduce the risk of polygenetic diseases.
Anne?
Yes.
Someone, I believe, misunderstood what you were saying. So use a stud dog without an NCL test if your bitch has been tested twice. I believe you said if you're going to breed to a carrier, test your bitch twice. And
well, yes, or a dog of unknown status, I would agree. Anytime you're going to a dog where they might have NCL, whether they're tested or they're not tested, I would test
your bitch twice and be sure she's clear.
That's what I would do.
Okay.
I know there's some people that are not testing dogs that are not from lines where it's been reported. But if you go on canine data and you search for the carriers, you will find some carriers that just show up and they are from backyard breeders from way back. So it's back there in our gene pool. I would not breed a golden retriever today unless I knew the puppies weren't going to have the disease. So complex polygenetic diseases, there are multiple genes involved. The environment may influence the disease. And we again look at some of the human genetic information, partly because it's in the public domain, so I can use the figures. But the other reason is because they're very well described. And so in terms of coronary artery disease, we know environment matters, but we know there are a number of risk genes, and they are on 22 of the chromosomes. And so these variants are your risk variants, and there are ways of coming up with scores that figure out what your risk is, but they are very complex, and we don't have it nearly this well mapped out in our dogs. And hip dysplasia in dogs is one of those diseases. There are multiple genes involved. The environment may influence the disease. And in a recent study of 1,600 dogs from 10 breeds, there were 21 chromosome sites on 14 different chromosomes that were found to affect hip dysplasia risk. 4 locations were significantly associated with risk in all breeds. This is not a simple autosomal recessive disease. There have been some DNA tests marketed for hip dysplasia. They didn't work very well. I think they've all gone off the market if they've been on for any length of time, because dogs got hip dysplasia from genes they weren't testing for.
And they probably looked at those 4 locations. And
actually, the 4 locations were just described in the past year.
Oh, wow.
Yeah, so this is very recent data. This
Okay.
is a 2020 study. Yeah. So other locations were associated with risks in specific breeds. The newest paper, the one in 2020, is identifying a potential common biochemical pathway, metallation, that may be involved. The road to hip dysplasia has many side roads that can get you to the same location. And this is the location for this AKC Canine Health Foundation write-up. on hip dysplasia and the complex genetics. Pigmentary uveitis, another big one that we think multiple genes and environment may be involved. It's an inflammatory disease of the eye of the golden retriever. It's very common in older golden retrievers. It can cause glaucoma, cataracts, and blindness. The pattern of inheritance is unknown, but it's believed to involve multiple genes. The average age of onset is 8.5 years of age, okay? That goes back to what the health and genetics, what the code of ethics says. Dogs that produce offspring should continue to have ophthalmology examinations on a yearly basis for their lifetime. And if the findings permit recertification, the results should continue to be recorded in an approved online database. This is not a calling out of the dogs that have pigmentary uveitis. This is, if something happens to you and your dog lived to be 14 and was free of the disease, someone may need to know about it when they're breeding to your dog's son, your dog's daughter. So that's really important.
And it's free after the dog is 8. It costs nothing to list it.
And for those seniors, once they hit that 8-year point, GRF will pay for it to be in that online database. So breeders can record success in breeding golden retrievers with healthy eyes for a lifetime. And as many breeders have recorded such success, all breeders can identify lines likely to have a lifetime of healthy eyes. We just want Goldens and their owners to be happy when their Golden gets older. And the online eye exam data can help breeders find the information that they need. And
well, what would you say to the person, because I see this all the time, who says, well, they can pick up the phone and call me if they want to know?
And I know people believe that, but I've lost so many friends. None of us are immortal. And I want to leave the world a better place. And I'm a veterinarian and I don't breed enough dogs to make a difference, but some of you have. And I'm very grateful to the breeders that have brought us to where we are today, where hip dysplasia is not where it was when I started in Goldens in the 1970s. And the whole idea is, yes, they can call you today, but what happens when you're gone? What we do as breeders should leave a legacy of health for the dogs of the future. So the DNA advisory recommendations for DNA tests are the prudent use of DNA testing can prevent disease in golden retrievers. NCL testing is extremely important for golden retriever breeding dogs because it is progressive and it is lethal. And what I want to ask everyone here to do is to promise me they will not let a puppy buyer receive a healthy puppy and watch it slowly die when that can be prevented. I don't want that happening. Please test for NCL. DNA testing is only helpful when a high quality testing laboratory conducts the test. I think I need to take Gusto out here, but we'll have to make this short. DNA testing for diseases that are not known to affect golden retrievers is not recommended because genetic background breed influences the effect of a DNA variant. The test for DM has some value, for example, but among veterinarians, if you start to talk to them about the DM test, they get livid because they've had clients euthanize dogs because they tested positive in breeds where the disease did not occur. And they felt they couldn't home the dog because it was going to die of the disease, and they euthanized the dog unnecessarily. So it's important. That is a test that Goldens can actually get, but we can't, most veterinarians want nothing to do with the DM test in breeds that don't usually get it because they hurt for the dogs that died, and they did. The counseling is much better these days. Many new DNA tests are anticipated in the future, and all golden retrievers likely carry some potential harmful recessive mutations. Automatic exclusion of carriers from breeding based on carrier status for recessive genes is not recommended. Genetic diversity is important for breed preservation. However, if you have a carrier for a serious disease, you must be transparent and responsible. Two carriers for the same fatal recessive disease should not be bred to each other, and we need to do what is necessary to prevent that from happening. And here are the resources and the references. We probably should post this at some point on Facebook.
We will do that.
Yeah, these are great references. The images, most of them, if I didn't draw them, they're coming from this website. It's a government website, so it's in the public domain. Our code of ethics, really a lot of thought went into writing the stuff on the clearances and what we do as responsible breeders. The Canine Health Foundation, the AKC Canine Health Foundation, has a wonderful, informative article that every breeder should read, and it was intended for breeders. It's a PDF, has nice illustrations. It's great. There's educational resources from the AKCHF on the complex genetic diseases that includes that information on the most recent article on hip dysplasia. And then some of the basic information on the diseases that can be a problem. There is good counseling data now for the DM test that's available from OFA. But as I said, enough veterinarians lost patients when the test first came out so that it's not popular in the veterinary community. And the stuff on the mitochondrial disease, that's the write-up, but this can all be posted on the web. And that's Gusto, who's in here, and I think he just ate a bunch of grass and got sick, so I need to go take care of him. But this is Gusto, and if anyone has any questions, I'm happy to answer the questions.
And I have an observation. You mentioned that many of us have dogs that are accomplished in various areas, and it's important for the breed that accomplished dogs who are healthy continue to be bred. It's interesting because Doug Johnson and Bill Shelton are doing a thing on dog preservation.
Yes.
And one of the things that they emphasized in their opening meeting was breeders need to be breeding dogs. I know it's harder for us now. We have less land, everything is expensive, and we keep fewer dogs, but we don't want to be our own bottleneck by just reducing and reducing and reducing the number of dogs we breed. Among other things, as you might take a look out 10 or 15 years, there will be very few good golden retrievers, well-bred golden retrievers who can contribute to the gene pool. So I know that politically we're not supposed to breed dogs anymore, but if we don't, then the dogs will be the result of puppy mill to puppy mill or backyard breeder to backyard breeder. So as
Yes.
Breeder Education Committee, we also want to encourage people to continue their breeding programs. Breed good dogs. Don't be shamed into thinking you can only have one litter every 3 years.
Oh,
Absolutely, absolutely, that's very true. And the more good breeders we have, the more diversity we have, and the more likelihood we are to have the dogs we need to pair up with for, the optimal cross. Not every stud dog is right for every female, and we don't all have to breed to the same stud dog, and we certainly shouldn't. And that's one thing used wisely, the genetic test can tell us is, hey, that dog is the #1 dog in the country, but not right for my girl.
Right.
And nothing against the #1 dog in the country. There's some great dogs, but it used to be that everyone wanted to go out and they wanted to breed to one dog. And that gives us a bottleneck. The same kind of thing can happen if we say we're not going to breed any carriers of a specific disease, particularly if the gene pool is limited.
And there's one question, and because I'm not the smart person on this, it says, wondering if Anne intends to discuss multiple generations of, quote, clear by parentage, unquote.
That is, the OFA will only register, I think, the first generation down as being clear by parentage. And a couple things that you just have to realize. Now with DNA testing, we have a much better idea of how many times the sire of the litter was not who the owner of the bitch thought sired the litter. It happens. And if somebody's kid let their dog out with their bitch and there's a litter of puppies and the breeder saw the breeding to the top stud dog in the country, they think the top stud dog in the country sired the litter, but maybe not. And it happens more often than you'd think. And so with that knowledge in hand, I think we have to realize that we need to test even if the grandparents were all clear. If the parents were clear and the kids got to go to college and you need every penny, if it's ichthyosis, okay. If it's NCL or CMS, be careful.
Not so much. Yeah.
That's just
so a rule of thumb might be one generation is your max.
There are some of it depends. I'm not going to tell someone if they've had the line and it's them and their spouse and there is no possibility of a problem and the grandkids going off to college. Again, it's not common in the line. Someone has a line where there's never been an NCL. Well, NCL would make me nervous, but but they're all coming down through clear stuff. I think it boils down to is what is the risk of a de novo, which is new in that individual mutation. Eventually you get to the point where you're trying to reduce a risk and a new mutation can happen. And when you start getting down to that risk, or the risk that the puppies are going to be damaged because the house gets struck by lightning. I mean, stuff happens. But I think breeders should use a degree of common sense, but they have to assure that puppy buyer that they're not handing them a healthy puppy that had a degenerative head NCL. Yeah. You can't do that. It's just, I do know people don't think it can happen to them because of the bloodline, but if you look at some of the NCL carriers, you would have never guessed it.
So we have what looks like a last question. Is there a test for cryptorchidism?
Not that I am aware of that is reliable. It's polygenetic. So as far as I know, there is no test. And I do periodically look at the tests that are coming out where there's something described for golden retrievers. And actually, one other thing I want to mention, and cryptorchidism is a good one for mentioning it on, is just because you have the disease and you have the gene doesn't mean that another gene can't cause the same disease, especially in another breed of dogs. So when I was in vet school, back when the dinosaurs roamed the earth, at that time, we thought that if your dog had been bred to a dog that had PRA, and the puppies were all clear, that your dog was test bred free of PRA and was of extreme value to the breed. We now know there are 20 different types of PRA. So you could have had a PRA carrier bred to a PRA affected, and that PRA affected might have had a different type of PRA, and your dog would have been test bred free. And that's where we've had to get away from the concept of they need to be normal clear. we thought, oh my gosh, it's a genetic disease. They're not normal. They have to be normal clear. Back when we thought PRA was one disease, but it's 20 diseases. So if you look at the number of diseases we have subsequently described, CMS, there's a golden retriever CMS, but there's a Labrador retriever CMS. And so, it's that way across all of these diseases. There's not one disease anymore. For PRA, there's 20, and for hip dysplasia, we know there are 22 chromosomes or something, that 14 chromosomes with 22 genes that can affect it. We can't have them all be normal clear for everything. That's not what we're seeking. We're seeking normal clear for hip dysplasia, normal clear for eyes, normal clears for elbows, normal heart exam from a cardiologist. That's normal clear. We're using DNA tests to prevent carrier puppies from being born. We're putting the examination results into an online database so that we can incorporate the genes from the healthy dogs. We're not calling out people that had a dog that wasn't normal clear. We all have dogs that aren't normal clear. If you have one that's normal clear for everything that's out there today, you just don't know the disease your dog is carrying.
Mm-hmm.
This, you have given us much more time than we told you we would take. We are, I think I speak for the whole Breeder Education Committee. Thank you so much for doing this for us.
I'm happy to do it.
And I'm sure it won't be the last one.
Well, thank you very much. I better get to Gusto. He's been fussing with me for quite a while.
And thanks to everyone who's attending. We'll do a couple things this time. We will put the entire webinar out on YouTube as we normally do, but we will also take the slide deck and make it available as just a slide deck, because that way you can also have all those resources that were at the end of the slide deck. So that might take us a couple days, but we'll get everything out there so people have access to this information and they can check back. So thank you all for
thank you,
for
Anne.
watching. We hope it was helpful.
Thank you.
Thank you. Good night, Gusto.
Thanks. Bye-bye.
Bye-bye.